Yes, dear scholars outside there, let us continue with our lesson of biology today. Today we are going to begin with the dihybrid test cross. Previously looked at the dihybrid cross. So right now, let us look at the dihybrid test cross. Strictly, I will say, consider a tall red flowered plant in the F2 generation of the dihybrid cross, and then we shall carry out a test cross on that plant. So I can write the question here that carry out a test cross. Carry out a test cross. Carry out a test cross on a tall red flowered plant. Tall red flowered plant in the F2 generation. In the F2 generation. In the F2 generation. F2 generation of the dihybrid cross. Of the dihybrid cross. So that is the question, dear scholars. That considering the dihybrid cross which we looked at previously, so carry out a test cross on a tall red flowered plant in the F2 generation. Now, I said a test cross is a cross of an organism or an individual bearing a dominant character with one that is homozygous recessive for a particular characteristic. So here we shall cross a tall red flowered plant with a plant which is homozygous recessive. So now in this case, the tall red flowered plant has two genotypes. It could be homozygous for tall or for red flowered, for red flowers, or it could be heterozygous. It could be heterozygous. So now, I will write solution. The tall red flowered plant, can either be homozygous, or heterozygous. So those are the two cases that we shall look at. Now, case number one, if the tall red flowered plant, is homozygous, then parental phenotypes. Parental phenotypes. We have tall, red, flowered plant, flowered plant. We are crossing it with short, white flowered plant. That is the first case. Crossing a tall red flowered plant with a short white flowered plant. So here we can see parental genotypes. These parents are haploid. Are diploid. The parents are diploid. So this one is homozygous for tallness and homozygous dominant for red flowers. We are crossing it with a short white flowered plant, genotype double recessive, that is small tt, and then small r. During meiosis, the genes segregate and then undergo independent assortment, as shown here on the board. So the genes will segregate. But of course, all allele combinations will be TR. All allele combinations in this parent will be TR. In a similar way, the possible allele combinations for the other parent, for the homozygous recessive parent, will also be TR, small t, small r. So I can write all small t, small r. So these alleles or these allele combinations are carried in gametes. They are carried in gametes which are haploid in nature. Now at fertilization, the genes, the gametes fuse, or the gametes combine together. So we can show our cross. We can draw the genetic cross then, so that we have random fertilization. Then the offspring will bear a genotype of T, capital T, small t, capital R, small r. So this is, we can say, testy cross genotype or offspring genotype. Offspring genotype, which are also 2n, since the diploid condition has been restored during the fertilization. Then we have capital T, small t, capital R, small r. So offspring phenotype. All tall, red flowered plants. All tall red flowered plants. So in the first case, that is what will happen when we cross a homozygous tall red flowered plant with a homozygous recessive short white flowered plant. So that is the first case. Case number 2 is when the parent of the individual with dominant characters is heterozygous. So I can write that if the tall red flowered plant, is heterozygous, then so if the other tall red flowered plant is heterozygous, of course we know that the other parent must be homozygous recessive. That is the meaning of a test cross. So we shall have parental phenotypes, parental phenotypes. We have tall and red flowered. Tall red. We are crossing it with short white. Then parental genotypes, of which the parents are diploid in nature. This parent is heterozygous, that is bearing a dominant gene, capital T, with a recessive gene, small t, then dominant R and recessive R. We are crossing it with still homozygous recessive, TT, small tt, small r, like that. That is where we are. As when the tall red flowered plant is heterozygous. So now, during segregation of the genes and independent assortment, then we shall have new allele combinations. Of course, this genotype here gives us 4 allele combination. The dominant T can combine with capital R within the same gamete to form an allele combination, capital T, capital R. Then the same dominant T can combine with this recessive allele, small r, to form an allele combination, capital T, small r. Also, that one is done. Now we go to the recessive allele for tallness, that is small t can combine with capital R to form an allele combination with alleles small t, capital R. And then lastly, small t, and small r are shown on the board. Those are the possible gametes. Now, whereas during segregation of the genes, for the genes of the homozygous recessive parent, all the allele combinations will be the same. They will be similar. They will all be small t and then small r. So I will simply write here, that all small t, small r as shown on the board. So we still maintain our cross sign here, even when gametes are crossing or are fusing together. All small t, small r. Now, these possible allele combinations can be carried in individual or separate gametes. So we can write gametes which are haploid in nature. Then we have meiosis here, meiosis during which segregation of the genes and independent assortment take place. So now, following that is now fertilization, which is random or random fusion of gametes. So each gamete here can potentially or randomly combine with the other gamete. So this is what we shall have. We shall have this gamete, cross it with this one here, then the other one also, this one here, will cross with the same gamete as shown on the board. Also, this allele, meaning this allele combination, will cross with the same gamete